delicate darlings, students. Okay, so this is Naza Zareen dealing with metabolism today. Metabolism is a process where it keeps the body in the living condition. So we are dealing with metabolism. It is a metabolic pathway, sequence of enzymatic reactions that are taking place in your body to keep your body in a surviving condition, that is to keep your body alive. So metabolism becomes very important in the body. So if you are going to take any kind of intake in your body, that is food, which becomes the primary source of energy because you need energy. So if you are consuming food, you either consume food in the form of biomolecules, that is either glucose or fructose, which are otherwise called as carbohydrates, or polysaccharides, or it is fatty acids, proteins, whatever it is. So irrespective of all these intake in the body through the diet, through the food, whatever is there, it has to break down itself to give you energy. If it has to give you energy, that means you will be in a state of living condition, you will be active enough. That proceeds with metabolism. Metabolism involves metabolic pathways. Metabolism is a process to keep your body in a living condition. So as I said, metabolism involves metabolic pathways. These metabolic pathways are catalyzed by various enzymes. So the sequence of enzymatic reactions become very important here. In metabolism, there are two types. One is anabolism and the other one is catabolism. I hope you can follow my writing. There are two types in metabolism. One is anabolism, the other one is catabolism. Anabolism is where synthesis takes place. Synthesis takes place. Catabolism is where breakdown takes place. Now, why should there be synthesis? Why should there be breakdown? Synthesis is required because you want to synthesize a high energy product with the precursors, the initiation molecules, and give you a high energy product. Suppose if you want energy, energy is usually in the form of ATP. ATP is the least energy currency molecule. Apart from ATP, you have high energy molecules, other high energy molecules like 1,3-diphosphoglycerate, phosphoenol pyruvate. These also give you energy, but you cannot use 1,3-diphosphoglycerate directly. You cannot use the phosphoenol pyruvate, otherwise called as PEP, directly. They have to break down into ATP. They go under serious metabolic reactions. That breakdown is nothing but catabolism. So anabolism is where synthesis takes place. Catabolism is where breakdown takes place. If you want to know in the most simpler terms, most of the people are suffering from diabetes mellitus. Diabetes mellitus is a state where the blood glucose level is high. That means there is glucose level high in the blood. So if glucose has to break down into glycogen, suppose the insulin comes into the picture. So in the presence of insulin, the glucose breaks down into glycogen. So that is nothing but the breakdown pathway. That is nothing but the catabolism. Suppose you need enormous energy to do various processes. You want the glucose back. Glucose is an energy. So you want the glucose back from the glycogen. If you have to get glucose back, it is synthesis. Simple terms, if you have glucose, it has to get converted to glycogen so that it maintains the blood glucose level in the body. That is known as catabolism. Okay? So that you don't suffer from what? Diabetes mellitus. Suppose if you want energy back, the glucose content is less in your body. So you go for the reverse reaction, will give you synthesis. So basically metabolism consists of anabolism and catabolism. Both are favorable to the body mechanism to keep the body in alive condition. Moving on to the next part, that is the various processes which takes place in the body. That is, as I said, metabolic pathways, as I said, sequence of enzyme reactions. So these metabolic pathways undergo the breakdown. Those I can term it as glycolysis, TCA cycle, and beta oxidation, and ETC, electron transport system. Okay, so if I am going to consider the various metabolic pathways in the body, I have four major. Okay, so we were dealing with metabolism. And under that, we were dealing with the various metabolic pathways in the body. The various metabolic pathways involves glycolysis, TCA, oxidation, beta oxidation, and ETC, that is otherwise called as ETS, electron transport chain or electron transport system. Before proceeding with these metabolic pathways, we'll have to know what exactly happens to the fate of the intake what we take in the body. That is the biomolecules like carbohydrates, polysaccharides, or fatty acids, otherwise called as lipids, or the proteins which are made from amino acids. Okay, these are the things which we take in our food, that is the biomolecules, which are carbohydrates or polysaccharides, fatty acids or lipids, proteins or otherwise amino acids. So what happens to the fate of these? All these will give you one major product which is otherwise called as acetyl-CoA. Acetyl-CoA, the heartbeat of metabolism. Okay, the central important molecule or substrate of metabolism. This acetyl-CoA actually enters into the TCA cycle. Irrespective of whatever it is, it enters into the TCA cycle, which is otherwise called as Krebs cycle. You get acetyl-CoA from beta oxidation pathway, you get acetyl-CoA from glycolysis, or you get acetyl-CoA from amino acid metabolism. That is what we were dealing. You get acetyl-CoA from glycolysis, you get acetyl-CoA from the beta oxidation, you get acetyl-CoA from proteins, which is otherwise one more amino acid metabolism. Okay, so you get acetyl-CoA. All this acetyl-CoA will enter into TCA cycle. This TCA cycle is all energy producing cycle. In this, there is oxidative enzymes are present. They undergo enormous, they give out high energy under oxidation. So they have enzymes which help in the production of energy. The enzymes will be dehydrogenases. The enzymes known as dehydrogenases, which catalyze the oxidation process. So enormous amount of energy is being liberated. This enormous amount of energy, whatever is being liberated, it is actually transferred to the coenzyme called NAD plus or FAD plus or FMN plus. These are coenzymes. So enzymes, with the help of enzyme dehydrogenase, the energy which is produced in the TCA cycle is being transferred to the coenzymes FMN plus, NAD plus, FAD plus. You should know what are coenzymes. Coenzymes are basically derived from the vitamins. If you are considering NAD plus, this is actually a derivative from vitamin niacin. If it is FMN plus or FAD plus, this is from the vitamin riboflavin. So these are coenzymes. This coenzymes, they take the reducing equivalents, that is from the substrate. Suppose if I am considering the substrate as A, let it be succinate, fumarate, malate, oxaloacetate, whatever it is, alpha ketoglutarate, glyceraldehyde 3-phosphate. So if I am considering this A as a substrate, okay, with the reducing equivalent, this reducing equivalent gets transferred to the coenzymes. Thereby, these coenzymes become reduced coenzymes. Reduced coenzyme in the sense, FMN plus becomes FMNH2, NAD plus becomes NADH plus H plus, and FAD plus will change to FADH2. Now, what is the funda of having all these reduced coenzymes? These reduced coenzymes have enormous amount of energy in it. We want energy in the form of ATP. ATP is an energy currency. So if you have one FMNH2, that means you have been provided with 2 ATPs. If you have one NADH plus H plus, that means it is giving you 3 ATPs. One FADH2 is going to give you 2 ATPs. So you are getting enormous amount of energy from these reduced equivalents or reduced coenzymes. So from the substrate, the reduced equivalent gets transferred to the coenzymes. From the coenzyme, it becomes a reduced coenzyme. This reduced coenzyme now has high energy in it. Further, you'll have to see these reactions. You'll have to know that FMN plus 2FMNH2 is a oxidation reaction. Okay. So obviously the enzyme will be dehydrogenase. The same way, the FAD plus gets converted to FADH2. This is again the oxidation process. The enzyme here will be dehydrogenase. The same way, NAD plus gets converted to NADH plus H plus. This is again the oxidation process and the enzyme here is dehydrogenase. Let us explain this with the example. Okay, we were speaking about the acetyl-CoA and then we discussed about the reducing equivalent, then the coenzyme and then reduced coenzyme and then the energy what you have got in the form of ATP. So, since the energy has come into the picture, let us deal with energetics of metabolism. Okay, how exactly we get energy? Energy should always be in the form of ATP and it should not be minus ATP. Minus ATP means it's consumption, it is utilizing the energy. So if it is the consumption of energy, it is thermodynamically unfavorable reaction. If it is giving out the energy, it is thermodynamically favorable reaction. So obviously in metabolism you see sequence of reactions either thermodynamically favorable or thermodynamically unfavorable, which you will know in the first pathway itself, that is glycolysis. So irrespective of whether it is endogonic process or exergonic process, both have to get coupled to each other. This coupling reaction keeps the body intact in the living condition. So dealing with the energetics, if you have to get ATP, as I said, from the substrate, the reducing equivalent has to be transferred. Suppose if I consider succinate, this is a metabolic substrate. This succinate gets converted to fumarate. Okay. So in this process, the substrate is getting converted to a new metabolite or new substrate, which becomes a substrate for the next one. That is, fumarate becomes substrate for the malate. Then malate becomes substrate for oxaloacetate. Okay. So succinate to fumarate. If I am considering the first step, that is succinate to fumarate. The enzyme here, since it is giving you energy, it should be succinate dehydrogenase. This is dehydrogenase enzyme because it is the conversion of FAD plus to FADH2. Okay, so this is succinate dehydrogenase because the process is oxidation. Okay, whenever oxidation comes into the picture, it is the dehydrogenase enzyme that comes into the picture. Which dehydrogenase? Substrate dehydrogenase. Since succinate is a substrate, it becomes succinate dehydrogenase. Suppose you have used pyruvate, then pyruvate dehydrogenase. Suppose you use lactate, lactate dehydrogenase. Suppose you use malate as a substrate, it becomes malate dehydrogenase, NADH. So depending on the substrate, you get dehydrogenase. The dehydrogenase is nothing but because of the oxidation process. Here FAD gets converted to FADH2. What has happened in the process is the substrate reacts in the presence of enzyme to give you fumarate, wherein another process is also taking place. So here these are coupled reactions. Another process where the proton which is present in the succinate is being transferred to the coenzyme. The coenzyme here is FAD+, which is actually derived from the vitamin riboflavin. You should remember that is being done previously. So this H2, the proton, is being transferred to the coenzyme FAD+, which gets converted to FADH2. So the reducing equivalent reduces the coenzyme further. This is coenzyme, now this is reduced coenzyme. This reduced coenzyme consists of enormous energy, and as we know, whenever FADH2 comes in the picture, it is plus 2 ATPs. So whenever succinate gets converted to fumarate, oxidation takes place and you get 2 ATPs. In the same way, if it is malate dehydrogenase, suppose I am taking this malate as my substrate and this malate gets converted to oxaloacetate. Okay, again this is a energy yielding step. Again this is giving you the reducing equivalent. So this is oxidation, this is MDH, malate dehydrogenase, but here the conversion will be NAD plus getting converted to NADH plus H plus. It is the niacin which comes in the picture. Here it was the riboflavin, but here it is niacin which comes in the picture. Hence NAD gets converted to NADH plus H plus. Thereby in this process you get 3 ATPs. So we have seen the conversion of FAD to FADH2 and we have seen the conversion of NAD to NADH plus H plus. Moving on to how to get energy directly in the form of ATP. There are certain reactions where the substrate doesn't need a coenzyme FAD plus. In the presence of the enzyme and in the presence of a cofactor, it directly gives you ATP. Such examples, you can consider the first step of glycolysis where glucose gets converted to glucose 6-phosphate. Okay, this step is known as phosphorylation. In this process, the ATP is being consumed. Actually, this is a energy consuming step. So ATP gets converted to ADP plus PI with the help of the cofactor Mg2+. And whenever phosphorylation comes in the picture, the enzyme used is kinases. The enzyme is kinases. Since we have seen that we want ATP, but here we are getting ATP is getting converted to ADP. Since ATP is being utilized, this is thermodynamically unfavorable reaction. Hence here directly he is taking out 1 ATP. So there is loss of ATP. Seeing the other reaction where that is again glycolysis. This is the first step of glycolysis. We take the last step of glycolysis where pyruvate is formed from phosphoenol pyruvate. So phosphoenol pyruvate gets converted to pyruvate. Here the ATPs, this is thermodynamically unfavorable, this is thermodynamically favorable. That's why ADP gets converted to ATP. Of course the enzyme here will also be kinases and the cofactor will be Mg2+. We have seen that here enzyme is there with a coenzyme, but here enzyme is there without a coenzyme but a cofactor to facilitate faster reaction. So as we know that the dehydrogenase will be respective substrate dehydrogenase, the same way kinases will be respective substrate kinase. So the entire name of the enzyme will be phosphoenol pyruvate kinases, glucokinases. So here we have got plus 1 ATP. So we have seen the energetics of metabolism where we have seen a thermodynamically unfavorable reaction, we have seen thermodynamically favorable reaction. We have seen direct formation of ATP or direct utilization of ATP, or we have seen the coenzymes getting reduced to give you the reduced coenzymes which further give you the respective ATPs. So this completes the energetics of the entire metabolism. Energetics which we were dealing now, let us just put that in a more simpler form in the form of a protocol or in the form of a tabular column. So the energetics of metabolism involves, if you are going to gain ATP, that means you have gained 1 ATP. If you have $1, that means you have $1 only. Okay? If you have gained it in GTP, then it is 1 ATP. Even GTP is being considered as 1 ATP itself. So this is direct phosphorylation giving you direct ATP. So the steps are otherwise called as phosphorylation. Okay, these two undergo phosphorylation step. Suppose if it is you have the reduced coenzymes like NADH plus H plus or FMNH2, and remember the next coenzyme, that is FADH2. So if you have NADH plus H plus, that means you have gained 3 ATPs. If you have FMNH2, that means you have gained 2 ATPs. If you have FADH2 again it is 2 ATPs. This energetic protocol becomes very necessary when you deal with energetics of different metabolic pathways. This step otherwise called as oxidation. We have dealt with phosphorylation, we have dealt with oxidation. So what is it possible that there will be reduction process? Of course, reduction process takes place when it is thermodynamically unfavorable. That means when anabolism takes place. So in anabolism, what happens is, instead of NAD getting converted to NADH plus H plus, instead of this, with the formation of 3 ATPs plus 3 ATPs, there are possibilities that you see a reverse reaction where NADH plus is getting converted to NAD plus. If it is in the forward direction, then it is oxidation. This is thermodynamically favorable, giving you plus 3 ATPs. If it is backward reaction, then it is reduction and it is loss of 3 ATPs. So, irrespective of whatever the reaction is, whether it is oxidation or reduction, you are either gaining ATP or you are losing ATP. Either oxidation is taking place or reduction. If oxidation is taking place, then it is substrate dehydrogenase. If it is the reduction taking place, the enzyme will be substrate reductase. Okay. So, if it is in the forward direction, you get plus 3 ATPs. If it is in the backward direction, you are getting minus 3 ATPs. Remembering these energetics, remembering this protocol, it is easy to plot energetics of any metabolic pathway. Starting on with the first metabolic pathway, that is glycolysis. So till now, whatever you have learned is actually the introduction to metabolism. As you proceed with the various metabolic pathways, things become more clear. Starting on with glycolysis. That is the conversion of glucose to pyruvate. The conversion of glucose to pyruvate. So glycolysis involves 10 steps, out of which it can be divided into 5. So it involves 10 steps. They are divided into 5 plus 5. The first 5 steps are nothing. They fall into preparative phase. And the next 5 steps fall into payoff phase. So glycolysis involves 2 phases, preparative phase and payoff phase. In preparative phase, it is actually energy consuming phase. In payoff phase, it is paying off the consumed energy along with enormous energy. So it is thermodynamically unfavorable and this is thermodynamically favorable. Further, in these 5 steps in preparative phase, the glucose gets converted to fructose, which further gets converted to fructose. Sorry, once again. Let me take a wider board phase, wherein glucose gets converted to glucose 6-phosphate, which gets converted to fructose 6-phosphate, which gets converted to fructose 1,6-bisphosphate, which gets cleaved further to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate is now eligible to enter the payoff phase. Yes. Stop. Okay, so the glucose gets converted to glucose 6-phosphate, which gets converted to fructose 6-phosphate, which gets converted to fructose 1,6-bisphosphate. Further, this gets cleaved to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. You'll have to know that glucose is a 6-carbon sugar. Glucose 6-phosphate again is a 6-carbon sugar. Fructose 6-phosphate 6-carbon. Fructose 1,6 is 6-carbon. But when a 6-carbon sugar cleaves to form 2 triose sugars because they are 3-carbon sugars. They get cleaved to triose sugars at the C3 and C4 cleavage. At C3 and C4 they cleave to get 3 carbon 2 sugars. This glyceraldehyde 3-phosphate now enters the payoff phase. Well, the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are actually isomers for each other. Okay, with the help of, since it is triose sugar, triose phosphate isomerase, the dihydroxyacetone phosphate can also get converted to glyceraldehyde 3-phosphate. So instead of the glyceraldehyde 3-phosphate alone entering into the payoff phase, the dihydroxyacetone phosphate can also get converted to glyceraldehyde 3-phosphate, and that also can enter into payoff phase. So instead of one glyceraldehyde 3-phosphate, you actually have two glyceraldehyde 3-phosphate. Hence, payoff phase is always multiplied by 2. Okay, this is very important when you deal with energetics. So I have divided this into preparative phase and payoff phase. The glucose, the first step of glycolysis, is otherwise called as phosphorylation. In this process, glucose gets converted to glucose 6-phosphate. That is, ATP gets converted to ADP. I said this is thermodynamically unfavorable reaction. So obviously it is consuming the high energy molecule ATP, which gets converted to ADP. So obviously it is known that there is loss of 1 ATP here. Since it is a phosphorylation step, and we know that kinase enzyme comes into the picture here. So the kinases will be glucokinase, the substrate kinase. It is glucokinase enzyme with the metal cofactor Mg2+. So the first step of glycolysis will be glucose getting converted to glucose 6-phosphate, where there is conversion of ATP to ADP plus Pi. So we know that whenever there is ATP, it gets converted to ADP plus Pi, or ADP plus Pi gets converted to ATP. Since here ATP is getting converted to ADP plus Pi, this is ATP, this is ADP. The Pi, the phosphate group, gets attached to the glucose at the 6th carbon. Hence it is known as, it is no more glucose now, it is glucose 6-phosphate. The glucose has a 6th carbon attached, the phosphate gets attached to the 6th carbon. So this is the first step of glycolysis. Moving on to the next step, that is glucose 6-phosphate gets converted to fructose 6-phosphate. This is just an interconversion step. Here, glucose and fructose are isomers of each other. Isomerism is a process where they share similar molecular formula, but they differ in their structures. So it is C6H12O6, this is also C6H12O6, but this has an aldehyde functional group, this has a keto functional group. This aldehyde functional group is at the carbon 1, this keto group has at the carbon 2. So that actually is an interconversion step, otherwise called as isomerization. Whenever isomerization comes in the picture, there is no loss or gain of energy. It's simple that glucose 6-phosphate gets converted to fructose 6-phosphate, which is a 6-carbon isomer. And this is not going to lose or gain energy. And the enzyme here will be isomerase. Which isomerase? Substrate isomerase. That is glucose 6-phosphate isomerase. Further, fructose 6-phosphate gets converted to fructose 1,6 bisphosphate. You can call it as fructose 1,6 diphosphate or fructose 1,6 biphosphate. That is, in the fructose 6-phosphate, the phosphate group is attached to the carbon 6. But here, the phosphate group not only get attached to carbon 6, it also has got attached to the carbon 1. So instead of one phosphate, it has two phosphates. Hence, it is called as bisphosphate, biphosphate, diphosphate, whatever it is fructose 1,6 diphosphate. Since the phosphate group is getting attached like previously, this step is also known as phosphorylation. We know that whenever phosphorylation comes in the picture, it is the kinases. So the enzyme here will be kinase. What kind of kinase? Fructose 6-phosphate kinase, substrate kinase. Here, since it is thermodynamically unfavorable reaction, you will have ATP getting converted to ADP, and the Pi has got attached to the first carbon. Of course, with the help of the metal ion thread Mg2+. So now, fructose 1,6 diphosphate, a 6-carbon sugar, gets cleaved to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Whenever cleavage comes in the picture, it is either aldolase, enolase, or whatever respective substrate is. Okay, so here since an aldehyde is since it is cleaving to 2 aldose sugars, the enzyme here will be aldolase, giving you dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Giving a quick review of preparative phase. Under glycolysis, glucose gets converted to glucose 6-phosphate, to fructose 6-phosphate, to fructose 1,6-bisphosphate, to dihydroxyacetone phosphate, to glyceraldehyde 3-phosphate. Glucose, the first step, phosphorylation, gets converted to glucose 6-phosphate, ATP gets converted to ADP, Pi gets attached here with the help of metal cofactor Mg2+, and the enzyme is glucokinase, loss of 1 ATP. Glucose 6-phosphate getting isomerized to fructose 6-phosphate in the presence of the enzyme glucose 6-phosphate isomerase. The step is known as isomerization. The second phosphorylation, what we see here, I have blocked one phosphorylation. This is the second phosphorylation what you see. Fructose 6-phosphate gets converted to fructose 1,6-diphosphate. ATP gets converted to ADP. Pi gets attached to the first carbon. Kinase enzyme, substrate kinase, fructose 6-phosphate kinase, Mg2+ as a cofactor. This 6-carbon sugar, a keto sugar, gets converted to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. It cleaves itself to 2 aldose sugars. Hence, the enzyme here is aldolase. There is no interconversion. There is no loss of energy. The total energetics of the preparative phase is -2 ATP. So, remembering this, okay, -2 ATPs, we will move on to payoff phase. We will be dealing with the glyceraldehyde 3-phosphate. Now, this glyceraldehyde 3-phosphate is ready to enter the payoff phase. So, the glyceraldehyde 3-phosphate in the payoff phase, it changes to 1,3-diphosphoglycerate. 1,3-diphosphoglycerate. This 1,3-diphosphoglycerate gets converted to 3-phosphoglycerate. 3-phosphoglycerate to 2-phosphoglycerate. 2-phosphoglycerate to phosphoenol pyruvate, giving you the end product, that is pyruvate. Glyceraldehyde 3-phosphate gets converted to 1,3-diphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate and pyruvate. In this process, since it has utilized 2 ATPs already, it wants to give out that 2 ATPs. It's trying to be a little loyal. So it tries to give out the 2 ATPs. Instead of giving 2 ATPs, it's a little happy and it would like to give out 3 ATPs. If it has to give 3 ATPs, obviously it has to undergo a reducing equivalent step. It has to give in the form of reduced coenzyme. Okay, so which is the reduced coenzyme which is going to give you 3 ATPs? That is NAD plus getting converted to NADH plus H plus. Okay, NADH plus H plus. So the reduced coenzyme here is NADH plus H plus. So it is giving you 3 ATPs. It is thermodynamically favorable phase. So glyceraldehyde 3-phosphate giving you 1,3-diphosphoglycerate, wherein NAD plus a coenzyme gets reduced coenzyme to NADH plus H plus, giving you 3 ATPs. In this process, since it is an oxidation step and since it is giving you ATP, it is no more just an oxidation, it is oxidative phosphorylation step. It is the oxidative phosphorylation. The enzyme here will be, whenever oxidation comes in the picture, the enzyme will be dehydrogenase, and the dehydrogenase enzyme is substrate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. So this I am going to block since this is an energy yielding step. 1,3-diphosphoglycerate gets converted to 3-phosphoglycerate. This is also a phosphorylation step. This 1,3-diphosphoglycerate, as I have told earlier in the introduction, we know that there are high energy substrates which give out ATP. So 1,3-diphosphoglycerate is a high energy substrate. Apart from that, you have phosphoenol pyruvate, which is a high energy substrate. Whenever phosphorylation takes place, when the high energy substrate breaks down, then it is known as substrate level phosphorylation. It is not just a simple phosphorylation, it is substrate level phosphorylation. Since we have 2 high energy compounds, that is 1,3-diphosphoglycerate and phosphoenol pyruvate, we can consider this as first substrate level level phosphorylation, and this as second SLP, substrate level phosphorylation. So we know this is a phosphorylation step. So obviously this is a energy consuming, energy producing step. So ADP gets converted to ATP. Whenever phosphorylation comes in the picture, it is the kinases. What kinase? 1,3-diphosphoglycerate kinase. So I am not going to write the complete name. It is the substrate kinase. Whenever kinase comes in the picture, it is the metal cofactor, your metal ion friend Mg2+. So it is gaining you 1 ATP. Okay. From 3-phosphoglycerate, you can take it as glyceric acid also. 3-phosphoglycerate, it gives you 2-phosphoglycerate. It is just the movement of the phosphate from the third carbon to the second carbon. That's all. This is known as, the enzyme here will be 3-phosphoglyceromutase. 3-phosphoglyceromutase, wherein the third carbon should, whatever phosphate was present in the third carbon, it has moved to the second carbon. So this is an interconversion step. From 2-phosphoglycerate, it gives a high energy substrate which is phosphoenol pyruvate. This, since we are getting enol form here, obviously there will be hydrolysis. So obviously the enzyme should be enolase. As we know, whenever the hydrolysis takes place, aldolase, enolase, fumarase. So obviously the enzyme, since it is giving a enol form, it is enolase. The phosphoenol pyruvate is a high energy substrate. This gets converted to pyruvate. So the step is otherwise called as phosphorylation. This is substrate level phosphorylation because high energy substrate is getting break down. And this is, since it is a thermodynamically favorable step, you get ADP gets converted to ATP, giving you +1 ATP. I have blocked this step. Of course, the enzyme will be kinase. What kinase? Substrate kinase, phosphoenol pyruvate kinase, with the metal ion friend Mg2+. So in this process, as I have blocked here, the energy currency, when we are dealing with the energy currency, we have seen that it has utilized 2 ATPs, -2 ATPs, but it wanted to give out +3 ATP. So it entered into dehydrogenase oxidative phosphorylation, giving you 3 ATPs. But since the preparative phase says I don't want the ATP in the form of the reducing equivalent, give me direct ATP. It enters the next step, substrate level phosphorylation, giving hit 1 ATP. Then again the preparative phase says you have given me 1 ATP, give me 1 more ATP. It says while going in the last step, I am going to give you your another 1 ATP. So in the last step, it again gives 1 ATP. So totally 3 + 1 + 1, that will be 5 ATPs will be the net currency of the payoff phase. So if you are going to consider this, we have seen that in the preparative phase, it was -2 ATP, and in the payoff phase, it is +5 ATP. We have told that it is not just one glyceraldehyde 3-phosphate entering, it is the dihydroxyacetone phosphate also getting converted to glyceraldehyde 3-phosphate, it also enters. So it is multiplied by 2, hence the payoff phase has to be multiplied by 2, giving you +10 ATPs. So +10 and -2, the total energetics will be 8 ATPs. So this is how we are going to calculate the energetics of glycolysis. Giving a quick review of payoff phase, glyceraldehyde 3-phosphate gets converted to 1,3-diphosphoglycerate to 3-phosphoglycerate to 2-phosphoglycerate to phosphoenol pyruvate to pyruvate. All these are 3-carbon sugars. Further, the glyceraldehyde 3-phosphate, it has to give out the ATP. It gives in the form of reduced coenzyme NAD to NADH + H+. Hence it is oxidation reaction. Since it is coupled with phosphorylation giving you ATP, it is otherwise called as oxidative phosphorylation. The enzyme will be dehydrogenase, substrate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. In this oxidative phosphorylation, it has given not only 3 ATPs, it has also given the high energy substrate 1,3-diphosphoglycerate. This 1,3-diphosphoglycerate enters into next, giving out 1 ATP, that is through substrate level phosphorylation. The enzyme here will be 1,3-diphosphogylucokinase. The cofactor will be Mg2+, ATP getting converted to ATP, giving you +1 ATP. 3-phosphoglycerate changes to 2-phosphoglycerate, just a slight conversion. From the third carbon, the phosphate moves to the second carbon with the enzyme substrate mutase. that is 3-phosphoglycerate mutase. Further, 2-phosphoglycerate undergoes hydrolysis in the presence of enolase. It gives a high energy substrate called as phosphoenol pyruvate. This phosphoenol pyruvate, which is a high energy substrate, undergoes substrate level phosphorylation, giving the end product of glycolysis, that is pyruvate. ADP gets converted to ATP. Of course, the enzyme is kinase, and the metal cofactor will be Mg2+, giving 1 ATP. So that completes preparative and payoff phase of glycolysis.